Hello, my name is Stephen Schur, and I'm a fifth-year MD PhD student at The Ohio State University. I will be presenting on VIP152, which is a novel selective CDK9 inhibitor and its role in chronic lymphocytic leukemia. I have no conflicts of interest to disclose, and the following authors have the following conflicts of interest. In brief, CLL is the most prevalent leukemia of adults in the Western world. It primarily affects the elderly population and is characterized by an expansion of clonal B cells. These malignant B cells are maintained through aberrant B cell receptor signaling. We have developed inhibitors of proteins downstream of the B cell receptor, notably Bruton's tyrosine kinase and the inhibitors ibrutinib and acalabrutinib. Additionally, positive clinical outcomes have been achieved with inhibitors of BCL2, the anti-apoptotic protein, with venetoclax. However, none of these inhibitors demonstrate curative therapies, and a significant portion of patients do develop resistance to these inhibitors, marking a need in the community for novel therapeutics. Additionally, venetoclax resistance has been shown to be mediated by MCL1 dependence, marking our movement into CDK9 inhibition. CDK9, in complex with one of its cyclin binding partners, forms the positive transcription elongation factor complex. This complex functions to phosphorylate RNA polymerase II and regulate short half-life transcripts, namely MYC and MCL1. Cyclin-dependent kinase inhibitors have been used in clinical trials. However, they often do not make it through the trials due to significant drawbacks of off-target related toxicity, thereby marking a particular need for novel selective therapeutics. To this end, we are investigating VIP152 and first looked into the selectivity of the compound. By performing a kinome scan, we were able to appreciate inhibition of CDK9 as the sole cyclin-dependent kinase at 100 nanomolar in a panel of 468 kinases. Second to CDK9 and its inhibition was GSK3 alpha, followed by ERBB2 and PIK5. When comparing VIP152's inhibitory capacity to other cyclin-dependent kinase inhibitors currently in development, we see that with a radiometric acellular ATP assay, we have increased potency and selectivity of VIP152 over dinaciclib, KB0742, and atiraciclib. Additionally, we do not observe any inhibitory capacity for VIP152 of CDK7 and cyclin H. We do appreciate increased inhibition of GSK3 alpha and GSK3 beta compared to these other inhibitors. We notice similar trends in the cellular NanoBrd-based assay. For VIP's 152 increased potency for CDK9 in cyclin T1, T2, and K. We do notice a small amount of inhibition for CDK7. However, this is to a lesser extent than dinaciclib and KB0742. When monitoring CDK9 activity as the phosphorylation markers of RNA polymerase II by Western blot, we appreciate in a CLL cell line treated with VIP152 at 1 micromolar for the listed time period, a time-dependent decrease in POLII phosphorylation at the second serine residue, a marker of CDK9 activity. Consequently, we notice a depletion of MCL1 and MYC protein levels as a function of time. Having now demonstrated the selectivity of our compound, we moved on to test the cytotoxic capacity. First, by performing an Exon-5 PI flow cytometry, we measured apoptosis of 2 CLL cell lines HG3 and MEK1 treated for 24 hours continuously with the listed doses of the compound and appreciated statistically significant decreases in viability. This trend was also maintained when looking at primary patient samples. 10 treatment-naive CLL patient samples were treated with the compound or vehicle control with or without the presence of stromal support, which has been shown to be a protective marker for CLL cells. We noticed that VIP152, when treated for 4 hours with 1 micromolar, the compound subsequently washed off, and then an Exon-5 PI flow cytometry performed at 24 hours, that we are able to overcome stromal protection to a significant extent. Having now demonstrated the cytotoxic capacity of our compound, we moved on to look at the transcriptomic effects given CDK9's intrinsic role in regulating transcription through the phosphorylation of POLII. We performed an experiment where we treated 4 primary CLL samples, which were treatment naive, for 2 hours with 1 micromolar of our compound. The cells were then sorted for live cells, which were CD45, CD5, and CD19 positive, and then sequenced according to an algorithm described in our lab. This principal component analysis demonstrates the robust separation of the 2 treatment groups, either vehicle or treatment with VIP152, along principal component 1, with each node representing the individual patient samples. These are the differentially expressed genes of this RNA sequencing experiment, and we do notice, as one would expect with CDK9 inhibition, that the majority of transcripts are downregulated, having been due to shutting down transcription. Notably, we do see downregulation of CDK9 axis transcripts, namely JUNB, MCL1, and BTG1. When we look functionally by pathway analysis and gene set enrichment analysis, we see an enrichment for cytoplasmic ribosomal transcripts in the treatment group, as well as an upregulation for transcripts belonging to the inhibition of rapidly degraded mRNA degradation pathways. Taken together, these data indicate a compensatory mechanism by which the cell is trying to increase translation as well as decrease RNA degradation as a result of having transcriptional machinery halted through CDK9 inhibition. Following this, we wanted to look into the protein-protein interactions affected by CDK9 inhibition. To accomplish this, we performed nuclear immunoprecipitation of CDK9 in a CLL cell line, which was treated for 2 hours with 1 micromolar of VIP152 or vehicle control, and then submitted this for proteomic analysis. This represents a small portion of the statistically significant differentially abundant proteins, which came out in our assay. We had previously observed that CDK9 and cyclin T1 were upregulated in the nuclear fraction when treated with our compound, consistent with previous data in flavopiridol. Additionally, we see a decrease in the abundance in the treatment group for transcriptional machinery, notably components of RNA polymerase II, RPB3, IWS1, and SPT6H, which are binding proteins to the C-terminal domain of the core component of RNA polymerase II. When we look functionally at the differentially abundant proteins, we see that they belong to gene expression, transcription, and RNA processing, as you would expect for proteins which associate with CDK9. In addition to this, we wanted to look at the changes in binding to the inhibitory 7sk RNA complex. By performing reciprocal co-IP with CDK9 and hexam1 in the nuclear fraction, treated with or without VIP152, we appreciate a decrease in association of P-TEFb with MEPCE, hexam1, and LARP7, all components of the 7sk RNA complex. This disruption of protein-protein interactions indicates a highly specific mechanism of action by which CDK9 is no longer capable of binding to its binding partners, albeit POLII for the transcription machinery or the inhibitory complex of the 7sk RNA. Having now looked mechanistically, we wanted to appreciate the in vivo capacity of our compound. So we performed an experiment where, using an in-house generated CLL-like model of the EMU MTCP1 overexpression transgenic mouse, we harvested splenocytes from the primary colony. 1 million splenocytes were engrafted via tail vein injection into Black 6 wild-type mice and monitored for peripheral disease by flow cytometry. The mice were randomly assigned to either VIP152 treatment or placebo and monitored for weekly flow cytometry of peripheral disease and overall survival. For the first 2 weeks following enrollment into treatment, mice which were given VIP152 demonstrated statistically significantly decreased peripheral disease compared to their vehicle counterparts. This translated into an improvement of overall survival for the treatment mice, with an improvement in median survival of 14 days. Having demonstrated that VIP152 functions as a monotherapy in CLL, we wanted to look into whether any synergy would be evident with inhibition of other genes. To accomplish this, we performed a CRISPR-Cas9 knockout screen using the Brunello library under the selective pressure of VIP152 or DMSO. This plot demonstrates the genes which came out of this screen. So the ones in blue on the left side are those genes whose knockout mediates synergy with VIP152, and the genes in red are the genes whose knockout mediates resistance to VIP152. Focusing on the synergy genes, when we perform pathway analysis on these genes, we notice that it is EIF2 signaling, which is the most enriched pathway, which follows with our upregulation of translation machinery that we noticed from our RNA-seq study. In conclusion, VIP152 is a selective CDK9 inhibitor with improved selectivity of other inhibitors in development. It disrupts binding of P-TEFb to canonical binding partners, thereby inhibiting its function. Treatment with VIP152 in the circulating adoptive transfer model of leukemia demonstrated a decrease of disease burden and improvement in survival. And these data are in support of an ongoing clinical trial of VIP152 in CLL.
These are my acknowledgements, and I thank you for your time today.